E4. Diversity of antibodies. E4. Diversity of antibodies. One unique property of antibodies is their remarkable diversity. Current estimates each human can synthesize more than 10, 11, 100 billion different kinds of antibodies. How is this diversity generated? The answer is threefold: rearrangement of antibody gene segments, somatic mutations, and generation of different codons during antibody gene splicing. Immunoglobulin genes are split or interrupted genes with many gene segments. Embryonic B cells contain a small number of gene segments, close together on the same chromosome, that determine the constant C region of the light chains, figure 1. Separated from them, but on the same chromosome, is a larger cluster of segments that determines the variable B region of the light chains. During B cell differentiation, one segment for the constant region is joined by a process of recombination mediated by specific proteins to one segment for the variable region. This splicing produces a complete light chain antibody gene. A similar splicing mechanism also occurs to join the constant and variable segments of the heavy chains. The light chain genes actually consist of 3 parts, and the heavy chain genes consist of 4. The formation of a finished antibody molecule is slightly more complicated than previously outlined, figure 2. Institute for Advanced Studies, Jodhpur 58. The germline DNA for the light chain gene contains multiple coding sequences called V and J joining regions. During the differentiation of a B cell, a deletion, which is variable in length, occurs that joins one V gene segment with one J segment. This DNA joining process is termed combinatorial joining because it can create many combinations of the V and J regions. When the light chain gene is transcribed, transcription continues through the DNA region that encodes for the constant portion of the gene. RNA splicing subsequently joins the VJ and C regions, creating mRNA. Combinatorial joining in the formation of a heavy chain gene occurs by means of DNA splicing of the heavy chain counterparts of V and J along with a third set of D diversity sequences, figure 3a. Initially, all heavy chains have a microtype of constant region. This corresponds to antibody class IgM, figure 3b. Another DNA splice joins the VDJ region with a different constant region that can subsequently change the class of antibody produced by the B cell, figure 3c. The amount of antibody diversity in the mouse that can be generated by combinatorial joining is shown in table 1. In this animal, the light chains can be formed from any combination of about 250 to 350 VK and 4JK regions, giving a maximum of approximately 1,400 different K chains. The lambda chains have their own V lambda and J lambda regions but smaller in number than their K counterparts, 6 different lambda chains. Heavy chains have approximately 250 to 1,000 VH, 10 to 30D, and 4JH regions, giving a maximum of 120,000 different combinations. Because any light chain can combine with any heavy chain, there will be a maximum of 2x108 possible K chain antibody types. The view of 2x108 different antibodies is actually an underestimate because antibody diversity is further augmented by 2 processes. 1. The V regions of germline DNA are susceptible to a high rate of somatic mutation during B cell development in response to an antigen challenge. These mutations allow B cell clones to produce antibodies with somewhat different polypeptide sequences. 2. The junction for either VJ or VDJ splicing and combinatorial joining can occur between different nucleotides and thus generate different codons in the splice gene. For example, a J splicing event can join the V sequence CCTCCC with the J sequence TGG in 2 ways: CCTCCCTGG, which codes for the amino acids proline and tryptophan, and CCTCCCTGG, which codes for proline and arginine. Thus the same VJ joining could produce polypeptides differing in a single amino acid. Institute for Advanced Studies, Jodhpur 59. E.5 Specific and non-specific immune response. The red body possesses 2 mechanisms which protect it from potentially dangerous viruses, bacteria, other pathogens, and abnormal cells which could develop into cancer. 1. One of these mechanisms is nonspecific, that is, it does not distinguish between infective agents. 2. The second mechanism is specific in that it responds in a very specific manner, e.g., production of antibodies to the particular type of infective agent. I. Nonspecific defense against infection. Nonspecific defense mechanisms help prevent entry and spread of invading microbes in an animal's body. Rightwards double arrow: an invading microbe must cross the external barrier formed by the skin and mucous membranes. Rightwards double arrow: if the external barrier is penetrated, the microbe encounters a second line of defense, interacting mechanisms of phagocytic white blood cells, antimicrobial proteins, and the inflammatory response. A. The skin and mucous membranes provide first-line barriers to infection. The skin and mucous membranes act as physical barriers preventing entry of pathogens, and as chemical barriers of antipathogen secretions. Rightward double arrow: in humans, oil and sweat gland secretions acidify the skin pH 3 to 5, which discourages microbial growth. Rightwards double arrow: saliva, tears, and mucus secretions contain antimicrobial proteins and wash away potential invading microbes. Rightwards double arrow: an enzyme, lysozyme, in perspiration, tears, and saliva attacks the cell walls of many bacteria and destroys other microbes entering the respiratory system and eyes. Right arrow: in the respiratory tract, nostril hairs filter inhaled particles and mucus traps microorganisms that are then swept out of the upper respiratory tract by cilia, thus preventing their entrance into the lungs. Rightwards double arrow: in the digestive tract, stomach acid kills many bacteria that enter with foods or those trapped in swallowed mucus from the upper respiratory system. B. Phagocytic cells, inflammation, and antimicrobial proteins function early in infection. 1. Microbes that penetrate the skin or mucous membranes encounter amoeboid white blood cells capable of phagocytosis or cell synthesis. Neutrophils are cells that become phagocytic in infected tissue. Neutrophils are produced in the bone marrow from the granulocyte monocyte stem cell. These cells are often called polymorphonuclear cells, PMNs. This is because of the polymorphic shape of the nucleus. Rightwards double arrow comprise 60%-70% of total white cells plays. Main role is in inflammation. Rightwards double arrow: they are the first cells to arrive at the site of inflammation by leaving the blood, through the endothelium into the tissue, extravasation. Rightwards double arrow: attracted by chemical signals, chemotaxis, they enter infected tissues by amoeboid movement. Rightwards double arrow only live a few days as they destroy themselves. When destroying pathogens, rightwards double arrow neutrophils are attracted into the tissue by chemotactic factors that include complement proteins, clotting proteins stimulated to be produced by tissue damage, and T-cell-derived cytokines. In the tissues, neutrophils are active phagocytic cells, like macrophages. Neutrophils, however, do not act as antigen-presenting cells. Neutrophils, instead, are most effective at killing ingested microorganisms and can do this by oxygen-dependent or independent pathways. Monocytes comprise only about 5% of the white blood cells, but they provide an even more effective phagocytic defense. They mature, circulate for a few hours, then migrate to the tissues where they enlarge and become macrophages. Macrophages are large amoeboid cells that use pseudopodia to phagocytize microbes, which are then destroyed by digestive enzymes and reactive forms of oxygen within the cell. Macrophages are long-lived. Rightwards double arrow most wander through interstitial fluid. Phagocytosing bacteria, viruses, and cell debris. Rightwards double arrow some reside permanently in organs and connective tissues. Rightwards double arrow fixed macrophages are especially numerous in the lymph nodes and spleen. Eosinophils represent about 1.5% of the total white cell count and have limited phagocytic activity. Rightwards double arrow contain destructive enzymes which are discharged against the outer covering of the invading pathogen. Eosinophils are granulocytes named because of their intense staining with eosin. Rightwards double arrow under the microscope. Eosinophils typically have a bilobed nucleus and contain many basic crystal granules in their cytoplasm. The granules are eosinophil mediators that are toxic to many organisms and also to tissues as in asthma. Rightwards double arrow eosinophils are motile and phagocytic and are particularly active in parasitic infection. In fact, blood work showing an increase in eosinophils is often caused by parasitic diseases. Rightwards double arrow main contribution is defense against larger invaders such as parasitic worms. Institute for Advanced Studies, Jodhpur 60. But they have similar importance in allergic reactions and are only found in tissues. Rightwards double arrow. There are two types of mast cells found either in the connective tissues or in mucosal sites. Both types contain numerous granules with preformed mediators which can be released from mast cells after stimulation. The preformed mediators include histamine and other pharmacologically active substances. Rightwards double arrow stimulation also results in the production of newly formed mediators by mast cells such as prostaglandins and leukotrienes. Stimulation of mast cells occurs in several ways such as by the anaphylotoxins C3a and C5a of the complement system or by the cross-linking of surface IgE. Mast cells have high affinity Fc receptors for the IgE that is produced against an allergen. As a result, mast cell release is most significant in either acute inflammation or in allergic responses. Natural killer NK cells destroy the body's own infected cells, especially those harboring viruses. These cells are sometimes called large granular lymphocytes, LGLs. NK cells have some surface markers in common with T cells, and they are also functionally similar to cytotoxic T lymphocytes, CTLs. Like CTLs, NK cells are particularly important in the killing of cellular targets, usually tumor cells. Unlike CTLs, however, the killing by NK cells is nonspecific. They do not need to recognize antigen MHC on the target cell. NK cells do not have a T cell receptor and are not T cells. An NK cell kills a target cell by releasing perforin and other molecules which damages the target cell membrane leading to death. NK cells also cause death by inducing apoptosis in the target. The cytokine TNF-alpha is released by the NK cells and may be involved in this process. Rightwards double arrow also assault abnormal cells that could form tumors. Rightwards double arrow are not phagocytic, but attack the membrane, causing cell lysis. 2. The inflammatory response. A localized inflammatory response occurs when there is damage to a tissue due to physical injury or entry of microorganisms. Rightwards double arrow vasodilation of small vessels near the injury increases the blood supply to the area, which produces the characteristic redness. Rightwards double arrow the dilated vessels become more permeable, allowing fluids to move into surrounding tissues, resulting in localized edema, swelling. Chemical signals are important in initiating an inflammatory response. Rightwards double arrow histamine is released from injured circulating basophils, type of white blood cell, and mast cells, type of cell found in connective tissue. Rightwards double arrow released histamine causes localized vasodilation, and the capillaries in the area become leakier. Rightwards double arrow increased blood flow to the site of injury delivers clotting elements that help block the spread of pathogenic microbes and begin the repair process. Migration of phagocytic cells into the injured area is also a result of increased blood flow and increased leakage from the capillaries. Rightwards double arrow phagocytes are attracted to the damaged tissues by several chemicals given. Rightwards double arrow neutrophils arrive first, followed closely by monocytes which develop into macrophages. Rightwards double arrow the neutrophils eliminate microorganisms and then die. Rightwards double arrow macrophages destroy pathogens and clean up the remains of damaged tissue cells and dead neutrophils. Rightwards double arrow dead cells and fluid leaked from the capillaries may accumulate as pus in the area before it is absorbed by the body. 3. Antimicrobial proteins. A number of proteins function in nonspecific defense by either directly attacking microorganisms or impeding their reproduction. The three most important nonspecific protein groups are complement proteins, interferon and the cytokines. The complement system is a group of at least 20 serum proteins that interact with other defense mechanisms. These proteins interact in a series of steps that result in lysis of the invading microbes. Complement activation is a cascade of events which resembles in some ways the coagulation pathway. It consists of a series of enzymes that become activated by each other in a cascade fashion. It's confusing and hard to remember, but still important because complement has an important role in clearing antigen. This is done by the four major functions of complement activation. Rightwards double arrow local vasodilation. Rightwards double arrow attraction of immune cells, especially phagocytes. Chemotaxis. Rightwards double arrow opsonization, or tagging, of foreign organisms. For phagocytosis. Rightwards double arrow destruction of invading organisms by the membrane attack complex MAC attack. The interferons are substances produced by virus-infected cells that help other cells resist infection by the virus. Rightwards double arrow they are secreted by infected cells as a nonspecific defense earlier than specific antibodies appear. Rightwards double arrow cannot save the infected cell, but their diffusion to neighboring cells stimulates production of proteins in those cells that inhibit viral replication. Rightwards double arrow not a virus-specific defense. Interferon produced to infection by one strain of virus produces resistance in cells to other unrelated viruses. Institute for Advanced Studies, Jodhpur 61. Rightwards double arrow most effective against short-term infections. Colds and influenza. Rightwards double arrow interferons are now being mass-produced using recombinant DNA technology and are being tested as treatments for viral infections and cancer. Cytokines are the chemicals produced by these cells in order to communicate and orchestrate the attack. Hormones in the endocrine system can produce an effect on other cells, so cytokines can act on other immune cells, especially cells that are close by. Cytokines have several important characteristics. Rightwards double arrow the same cytokine may be made by a number of different cells. Rightwards double arrow the same cytokine may have different effects in different circumstances. This is called pleiotropy. Rightwards double arrow different cytokines may have the same activity depending on the situation. Rightwards double arrow cytokines often act together and increase the effects of one another. Synergy. Rightwards double arrow they may also act as antagonists. Rightwards double arrow most cytokines have either paracrine or autocrine effects. Paracrine means they act on cells near to them or that they are actually touching. different function of IL-2 is well known because, when a T cell is stimulated to make IL-2, it stimulates itself via the IL-2 receptor to proliferate. An example of an uncommon endocrine function for cytokines is IL-1, which can cause fever by stimulating the hypothalamus. 2. How specific immunity arises. The adaptive immune system is the body's third line of defense and is very specific in its response. It is distinguished from non-specific defenses by specificity and diversity. Non-self recognition memory. Specificity refers to the immune system's ability to recognize and eliminate particular microorganisms and foreign molecules. Lymphocytes provide the specificity and diversity of the immune system. Lymphocytes, a type of white blood cell, are responsible for both humoral and cell-mediated immunity. The different responses are due to the two main classes of lymphocytes in the body, B cells and T cells. Rightwards early B and T cells, as well as other lymphocytes, develop from stem cells in the bone marrow and are very much alike. They only differentiate after reaching their site of maturation. Rightwards B cells, B lymphocytes, are responsible for the humoral immune response. Rightwards they form in the bone marrow and remain there to complete their maturation. Rightwards T cells, T lymphocytes, are responsible for the cell-mediated immune response. Rightwards they also form in the bone marrow, and then migrate to the thymus gland to mature. Diversity refers to the immune system's ability to respond to numerous kinds, millions, of invaders, which are recognized by their antigenic markers. Each population of antibody-producing lymphocytes is stimulated by a specific antigen. The stimulated lymphocytes synthesize and secrete the appropriate antibody. Antigens interact with specific lymphocytes, inducing immune responses and immunological memory. The ability of the immune system to respond to the wide variety of antigens which enter the body is based in the enormous diversity of antigen-specific lymphocytes present in the system. Each lymphocyte will recognize and respond to only one antigen. This specificity is determined during embryonic development before any antigens are encountered, and is the consequence of the antigen receptor on the lymphocyte's surface. When an antigen enters the body and binds to receptors on the specific lymphocytes, those lymphocytes are activated and begin to divide and to differentiate. Rightwards the divisions produce a large number of identical effector cells, clones, which bind to the antigen that stimulated the response. For example, a B cell is activated, it will proliferate to produce a large number of plasma cells that will each secrete an antibody that functions as an antigen receptor for the specific antigen that activated the original B cell. Thus, each antigen activates only a small number of the diverse group of lymphocytes. The activated cells proliferate to produce a clone of millions of effector cells which are specific for the original antigen. Clonal selection. Clonal selection equals antigenic specific selection of a lymphocyte that activates it to produce clones of effector cells dedicated to eliminating the antigen that provoked the initial immune response. One clone of cells consists of effector cells. Another clone of cells consists of memory cells. Rightwards effector cells are the short-lived cells that actually defend the body during an immune response. Words effector cells are populations of cells resulting from division of lymphocytes that were activated by the binding of antigens to their antigen receptors. Institute for Advanced Studies, JODPR 62. E6. Interaction responsible for antigen-antibody interaction. Strength of antigen-antibody interactions. The covalent interactions that form the basis of antigen-antibody (AGAV) binding include hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals interactions. Figure 1. Because these interactions are individually weak compared with a covalent bond, a large number of such interactions are required to form a strong Ag-A flat interaction. Each of these non-covalent interactions operates over a very short distance, generally about 1 x 10 to 7 1 angstrom. Consequently, a strong AGAV interaction depends on a very close fit between the antigen and antibody. Such fits require a high degree of complementarity between antigen and antibody, a requirement that underlies the exquisite specificity that characterizes antigen-antibody interactions. Antibody affinity is a quantitative measure of binding strength. The combined strength of the non-covalent interactions between a single antigen binding site on an antibody and a single epitope is the affinity of the antibody for that epitope. Low-affinity antibodies bind antigen weakly and tend to dissociate readily, whereas high-affinity antibodies bind antigen more tightly and remain bound longer. Association between a binding site on an antibody A flat with a monovalent antigen Ag can be described by the equation. Antibody avidity. The affinity of one binding site does not always reflect the true strength of the antibody-antigen interaction. When complex antigen multiple, repeating antigenic determinants are mixed with antibodies containing multiple binding sites, the interaction of an antibody molecule with an antigen molecule at one site will increase the probability of reaction between those two molecules at a second site. The strength of such multiple interactions between a multivalent antibody and antigen is called the avidity. The avidity of an antibody is a better measure of its binding capacity within biological systems, e.g., the reaction of an antibody with antigenic determinants on a virus or bacterial cell, than the affinity of its individual binding site. High avidity can compensate for low affinity. For example, secreted pentameric IgM often has a lower affinity than Ag, but the high avidity of IgM, resulting from its higher valence, enables it to bind antigen effectively. Cross-reactivity. Ag-flat reactions are highly specific. In some cases, antibody elicited by one antigen can cross-react with an unrelated antigen. Such cross-reactivity occurs if two different antigens share an identical or very similar epitope. In the latter case, the antibody's affinity for the cross-reacting epitope is usually less than that for the original epitope. Cross-reactivity is often observed among polysaccharide antigens that contain similar oligosaccharide residues. The ABO blood group antigens, for example, are glycoproteins expressed on red blood cells. Subtle differences in the terminal residues of the sugars attached to these surface proteins distinguish the A and B blood group antigens. An individual lacking one or both of these antigens will have serum antibodies to the missing antigen. The antibodies are induced not by exposure to. Institute for Advanced Studies, JODPR 63. Red blood cell antigens but by exposure to cross-reacting microbial antigens present on common intestinal bacteria. These microbial antigens induce the formation of antibodies in individuals lacking the similar blood group antigens on their red blood cells. In individuals possessing these antigens, complementary antibodies would be eliminated during the developmental stage in which antibodies that recognize self-epitopes are weeded out. Antibodies, although elicited by microbial antigens, will cross-react with similar oligosaccharides on foreign red blood cells, providing the basis for blood typing tests and accounting for the necessity of compatible blood types during blood transfusions. A type A individual has anti-B antibodies, a type B individual has anti-A, and a type O individual thus has anti-A and anti-B. A number of viruses and bacteria have antigenic determinants identical or similar to normal host cell components. In some cases, these microbial antigens have been shown to elicit antibody that cross-reacts with the host cell components, resulting in a tissue-damaging autoimmune reaction. The bacterium Streptococcus pyogenes, for example, expresses cell wall proteins called M antigens. Antibodies produced to streptococcal M antigens have been shown to cross-react with several myocardial and skeletal muscle proteins and have been implicated in heart and kidney damage following streptococcal infections. ESN, primary and secondary immune are ESPONE. The primary immune response is the proliferation of lymphocytes to form clones of effector cells specific to an antigen during the body's first exposure to the antigen. Rightwards double arrow. There's a 10 to 17 day lag period between initial exposure and maximum production of effector cells. Rightwards double arrow. The lymphocytes selected by the antigen are differentiating into effector B cells and T cells during the lag period. Rightward double arrow. Activated B cells give rise to effector cells called plasma cells, which secrete antibodies humoral response that eliminate the activating antigen. A secondary immune response occurs when the body is exposed to a previously encountered antigen. Rightwards double arrow. The response is faster, 2 to 7 days and more prolonged than a primary response. Rightwards double arrow. The antibodies produced are more numerous, and they are more effective at binding the antigen. This ability to recognize a previously encountered antigen is known as immunological memory. Rightwards double arrow. Based on memory cells, which are produced during clonal selection for effectors in a primary immune response. Rightwards double arrow. Memory cells are not active during the primary response and survive in the system for long periods. Effector cells produced in the primary response are active, and thus, short-lived. Rightwards double arrow. When the same antigen that caused a primary immune response again enters the body, the memory cells are activated and rapidly proliferate to form a new clone of effector cells and memory cells. Rightwards double arrow. These new clones of effector and memory cells are the secondary immune response. Rightwards double arrow. This acquired immunity has long been recognized as a resistance to some infections encountered earlier in life, e.g., chickenpox. Lymphocyte development gives rise to an immune system that distinguishes self from non-self. Rightwards double arrow. Like all blood cells, lymphocytes derive from stem cells in bone marrow or liver of a developing embryo. Initially, all lymphocytes are alike, but depending on the site of maturation, they develop into T cells or B cells. Rightwards double arrow. Cells that remain in the bone marrow develop into B cells. The B stands for bursa of Fabricius, an organ found in birds where they were first discovered. However, you can equate the B with bone. Rightwards double arrow. Cells that migrate to the thymus develop matures into T cells. The T stands for thymus. Immune tolerance for self. Self-non-self recognition is the ability of the immune system to distinguish between the body's own molecules and foreign molecules, antigens. Rightwards double arrow develops before birth when T and B lymphocytes begin to mature in the thymus and bone marrow of the embryo. Rightwards double arrow. Antigen receptors on the surfaces of lymphocytes are responsible for detecting foreign molecules that enter the body. There are no lymphocytes reactive against the body's own molecules under normal conditions. Rightwards double arrow. Self-tolerance equals the lack of a destructive immune response to the body's own cells. Rightwards double arrow. Failure of this system leads to autoimmune disorders that destroy the body's own tissues. Institute for Advanced Studies, Jodhpur 64. E8. The complement components. Protein glycoproteins that compose the complement system are synthesized mainly by liver hepatocytes, although significant amounts are also produced by blood monocytes, tissue macrophages, and epithelial cells of the gastrointestinal and genitourinary tracts. These components constitute 5% by weight of the serum globulin fraction. Most circulate in the serum in functionally inactive forms as proenzymes, or zymogens, which are inactive until proteolytic cleavage, which removes an inhibitory fragment and exposes the active site. The complement reaction sequence starts with an enzyme cascade. Common components are designated by numerals C1, C9, by letter symbols e.g., factor D, or by trivial names e.g., homologous restriction factor. Peptide fragments formed by activation of a component are denoted by small letters. In most cases, the smaller fragment resulting from cleavage of a component is designated A, and the larger fragment designated B, e.g., C3a, C3b. Note that C2 is an exception. C2a is the larger cleavage fragment. The large fragments bind to the target near the site of activation, and the smaller fragments diffuse from the site and can initiate localized inflammatory responses by binding to specific receptors. The complement fragments interact with one another to form functional complexes. Those that have enzymatic activity are designated by a bar over the number or symbol e.g., C4b2a, C3bbb. Complement activation. Figure 1 outlines the pathways of complement activation. The early steps, culminating in formation of C5b, can occur by the classical pathway, the alternative pathway, or the lectin pathway. The final steps that lead to a membrane attack are the same in all pathways. The classical complement pathway. Activation of the classical complement pathway requires initiation by the interaction of antibodies with an antigen that is usually cell-bound. Figure 1. The order of effectiveness in activating complement is as follows: IgM Ig3 Ig1 Ig2. However, some microbial products, lipid of endotoxin and staphylococcal protein A, or plasmin, a proteolytic enzyme that dissolves the fibrin of blood clots, may activate C1 directly without antibody participation. Following binding of antigen to antibody, the C1 complement component, which is composed of 3 proteins Q, R, and S, attaches to the Fc portion of the antibody molecule through its C1Q subcomponent. In presence of calcium ions, a trimolecular complex C1QRS Ag A flat that has esterase activity is rapidly formed. The activated C1 subcomponent attacks and cleaves its natural substrates in serum C2 and C4. Leads to binding of a portion of each molecule, C2b and C4b, to the antigen-antibody complement complex with the release of small C4a and C2a fragments. The released C2 fragment traditionally has been called C2a. For consistency, this text will label all larger complement fragments that are bound to the target cell as B fragments. With the binding of C2b to C4b, an enzyme with trypsin-like proteolytic activity is generated. The natural substrate for this enzyme is C3, thus it is termed AC3 convertase. Activity of C4b2b, the bar indicates an active enzyme complex. C3 is cleaved into a bound subcomponent C3b and AC3A soluble component. C3b then absorbs to bound C4b2b, forming the complex C4b2b3b, which acts as AC5 convertase and cleaves C5 into fragments C5a and C5b. Lysins 7 rapidly bind to C5b, forming a C5b67 complex that possesses an unstable membrane binding site. Once bound to a membrane, this complex is stable. C and C9 then bind, forming the membrane attack complex C5b6789 that creates a pore in the plasma membrane of the target cell. It is believed that the actual pore is a donut-shaped polymer of C9. The proforin pores generated by cytotoxic T lymphocytes are somewhat similar to complement pores. If the cell is eukaryotic, Na and H2O enter through the pore leading to osmotic lysis of the cell. If the cell is a gram-negative bacterium, lysozyme from the blood enters through the pore and digests the peptidoglycan cell wall, causing the bacterium to lyse osmotically. In contrast, gram-positive bacteria are resistant to the cytolytic action of the membrane attack complex because they lack an exposed outer membrane and the thick peptidoglycan prevents an attack on the plasma membrane. Institute for Advanced Studies, Jodhpur 65. E9. Major histocompatibility complex HLA in humans. The major compatibility complex MHC, sometimes referred to as human leukocyte antigens or HLA in humans, is a group of glycoproteins proteins with attached sugar chains embedded in the plasma membranes of cells. T cells are the antigen that come form the inside of the cells. They use an antigen recognition system which is distinct from the antigen recognition system. The T cell receptor TCR for antigen interacts with the antigen almost exclusively only when fragment of antigen is on surface of cell and bound to a molecule known as MHC gene product. Thus the role of MHC molecules coded by the MHC is to bind to peptide fragments derived from the protein antigens and interact with TCR. The binding of MHC molecule with peptide is selective. Arrow MHC molecules may be viewed as third set of recognition molecules for the antigen and the immune response, in addition to antigen-specific T cells and B cells. Institute for Advanced Studies, Jodhpur 66. Rightwards double arrow. Every vertebrate species has MHC genes and products which are cause of rejection of tissues between different members of same species and are dominated by T cells. Rightwards double arrow. These important self-markers are coded for by a family of genes. Rightwards double arrow. There are at least 20 MHC genes and at least 100 alleles for each gene. Rightwards double arrow. This results in millions of different MHC molecules. Rightwards double arrow. The probability that two individuals will have matching MHC sets is virtually zero unless they are identical twins. There are two main classes of MHC molecules in the body. Class 1 MHC molecules are located on all nucleated cells of the body. Class 2 MHC molecules are found only on specialized cells, such as macrophages, B cells, and activated T cells. Although MHC molecules give identity to the cell as being self, the major function of MHC molecules is to bind to an antigen, then present this antigen on the surface of the cell, thereby facilitating antigen binding to a T cell. Rightwards double arrow. Class MHCI molecules facilitate antigen binding to cytotoxic T cells, CD8 cells. Rightwards double arrow. Class MHC2 molecules facilitate antigen binding to helper T cells, CD4 cells. Each MHC antigen combination forms a unique complex that is recognized by specific antigen receptors on certain T cells. Humoral and cell-mediated immune responses. The body will mount either a humoral response or a cell-mediated response, depending on the antigen which stimulates the system. Rightwards double arrow. Humoral immunity produces antibodies in response to toxins, XV venom, free bacteria, and viruses present in the body fluids.